Academy. This channel is Academy Future Doctors. I'm your educator in biochemistry, Dr. Shivangi Shukla. I welcome all of you to the third session for tonight, that is, yeah. Here we shall be discussing about the important points regarding the urea cycle and how urea is metabolized in how ammonia is metabolized inside our body and in the form of urea. But first, let me just check if audio is clear and if this is smoothly. I can see that the audio and the video are proper. Okay. So, let me just quickly talk about the different types of subscriptions for NEET that are available on the Unacademy App. There are two types of subscriptions, Iconic as well as Plus. Now, both of them provide you access to video lectures from top educators across the country, the live quiz that they conduct, and question bank containing 5,000 questions. Also, their printed PDF notes that the educators provide, so that you don't waste time or you focus mainly upon developing class rather than focusing on taking down. Okay. And the Iconic subscription will also provide you with access to resources from Preplatterers. Do not forget to put in the educator code DRSHULA Dr. Shukla to avail an additional 10% discount. The discount is DSHULA Dr. Shukla for an additional 10% discount over and above the already subsidized prices. Okay. Now, we are starting with the urea cycle, but first of all, let us see what is urea. Okay, so urea is the product of ammonia, metabolism of ammonia. Now, how is ammonia produced? Now, look at the biomolecule called protein. Proteins have a carboxy terminal and an amino terminal. They are made up of amino acids having a carboxy terminal and an amino terminal. Okay. Now, once they are degraded or digested inside our body, what do they produce? They produce ammonia. Now, this ammonia is highly toxic. Okay, it is highly toxic. It is conjugated by glutamate or glutamine. Okay, it is conjugated by this, so that free ammonia does not roam about does not wander in the blood and cause toxicity. Okay, and this ammonia will have to be excreted out of the body. It cannot be excreted as such because it is toxic. So there are three types of organisms depending upon how they excrete ammonia, which is a waste product of what? Waste product of protein metabolism. Understanding this? So depending upon the mode of excretion of ammonia, the organisms can be either a monotelic, that means they excrete ammonia as such, like fish. Okay. Second, they can be either ureotelic, that is, they excrete ammonia in the form of urea, like man. Third, they can be either uricotelic. What is uricotelic? They will excrete ammonia in the form of uric acid, like birds. Very important. Okay. Now, uric acid utilizes very little amount of water. Therefore, that is what birds do. They secrete uric acid pellets. Okay. And that is their mode of excretion of ammonia. There are three types of organisms depending on how in which form they excrete. They can be either a monotelic or ureotelic or uricotelic. Now we are concerned with ureotelic organisms in our lecture here. Okay, so let us discuss how urea is synthesized. Now it is synthesized in compartments, that is, one is the mitochondria and second is the cytosol. There are total 5 important reactions occurring, and these reactions, the first 2 reactions are occurring inside the mitochondria and the last 3 are occurring inside the cytosol. And what is the important site where it is occurring? Always remember, the site for synthesis is mainly the liver, from where urea is excreted through the kidneys, and a small amount of urea formation also occurs in the kidney. Understood? So if serum urea is less, that means there is something related to the liver. That means the liver, the synthetic property of liver is getting affected. Liver has two major functions: synthesis and clotting. Isn't it? Clotting factors and synthesis of different factors. So synthetic function is when different proteins that it synthesizes, they get affected. So here in the liver, when suppose the urea, whenever serum urea is less, it can either be that the diet is very deficient, extremely deficient in protein, so ammonia is not getting synthesized in a large enough, and urea is lesser. Or there is, the person is suffering from dilution of the blood. There's a lot of water, because of which every factor is getting decreased because of hemodilution. And third thing is, because of some problem in the liver, the urea is not getting synthesized. So there will be symptoms of encephalopathy. Why encephalopathy? Because ammonia that is getting excess in the blood, excess of ammonia, because it is not getting converted to urea, that excess of ammonia is going to affect the blood-brain barrier, get inside the brain, and it is going to produce symptoms of disorientation and loss of consciousness, disorientation, and a lot more other symptoms that are significant of encephalopathy. So this is how you differentiate between the different causes of low serum urea. High serum urea is mostly due to either acute kidney injury or chronic kidney. Understood? Or due to a diet that is extremely rich in proteins, or in cases of dehydration. Understood? Got it? So now we'll begin with the urea cycle. Let us have an overview. Carbon dioxide and ammonium ions, they will combine in the presence of the enzyme carbamoyl phosphate synthetase. Okay, carbamoyl phosphate synthetase 1, CPS1. Since it is synthetase, that means it will utilize ATP, and it will have magnesium as the cofactor. What will it synthesize? It will synthesize carbamoyl phosphate. Okay, so carbon dioxide and ammonia will, in the presence of carbamoyl phosphate synthetase 1, it will form carbamoyl phosphate. Okay, now what will happen? This carbamoyl phosphate is going to catalyze the conversion of ornithine to citrulline, utilizing the enzyme OTC, ornithine transcarbamylase, OTC. So this is the first reaction catalyzed by carbamoyl phosphate synthetase 1, and the second reaction is catalyzed by ornithine transcarbamylase. Both of these reactions occur in the mitochondria. Now this citrulline exits the mitochondria and enters the cytosol. Inside the cytosol, it is acted upon by arginosuccinate synthetase. That means it is utilizing energy again. Synthetase means it will synthesize arginosuccinate, as you can see here. Then there's the enzyme arginosuccinase. Succinase ase means it is going to break it into arginine and fumarate. This fumarate is going to go where? Can I get the answer? This fumarate is going to go in which cycle? Where have you heard of it? Where have you heard of fumarate? Try and recall where you have heard of fumarate. This fumarate is going to go to the Krebs cycle, okay, or the TCA cycle. Understood? And this arginine is then going to follow the last step, that is arginase. That means breakdown of arginine. to produce urea, and then again it is giving ornithine, and the cycle is continuing. Understood this? So this, just look at the urea cycle once and then you understand how this is. Okay. How is it starting? The first step is carbamoyl synthase 1 and carbamoyl phosphate synthetase 1 enzyme that is going to convert carbon dioxide and ammonium into carbamoyl phosphate, and then this carbamoyl phosphate is going to catalyze the conversion of ornithine to citrulline. This citrulline will then leave, and what is the enzyme? It is ornithine transcarbamylase. Now this this citrulline will leave the mitochondria and enter the uh, cytosol. Sorry. And in the cytosol, again, it will be subjected. There will be 3 steps. Citrulline converting to arginosuccinate. What is the enzyme? Arginosuccinate synthetase utilizing ATP. Arginosuccinate will then convert to arginine and fumarate using arginosuccinase. Arginine will then break down again to ornithine using arginase. Okay. And what is it going to produce? It is going to produce urea. Understood this or not? So this is how urea is produced. Okay. And this important enzyme, carbamoyl phosphate synthetase enzyme, it is regulated by what? It is regulated by a compound that is N-acetyl glutamic acid, which allosterically activates carbamoyl phosphate synthetase 1. Understood? N-acetyl glutamic acid. Got it or not? Okay. And what was the amino acid that was combining, conjugating with ammonia? That was glutamate. Okay. Glutamate. N-acetyl glutamic acid is going to allosterically activate carbamoyl phosphate synthase 1, and glutamate is going to combine with ammonia to prevent free ammonia from getting transported into the blood and causing toxicity. Okay, understood this or not? So this is regarding the urea cycle. Okay, and all regarding the urea cycle. You can look at this, all the important steps that are occurring. The first 2 steps are occurring in the mitochondria. The last 3 steps are occurring in the cytosol. Okay, got it? So I think you have understood the process of urea cycle now. If there's any doubt that you guys have, you can ask me in the chat section. Otherwise, I think we can conclude this session. Let us just revise it again. And how is it linked to the Krebs cycle? It is linked through fumarate. Okay, it is linked through fumarate. Fumarate links the Krebs cycle and the urea cycle together. Okay, how? When it will move from the urea cycle to the Krebs cycle. Also, there is this intermediate, that is oxaloacetate, that is present where, if they ask you what is the one substrate that links urea cycle to Krebs cycle, you will say it is fumarate. And if they ask you what is this one amino acid that links urea cycle to Krebs cycle, then you will answer aspartate. And why is it so? Because aspartate is formed through transamination of oxaloacetate, and this aspartate is going to donate one of its, this oxaloacetate is present in the Krebs cycle, and this aspartate is going to donate one of its ammonia here, one of its nitrogen atoms here. Understanding or not? Very, very important. So one amino acid that is linking urea cycle and Krebs cycle is aspartate. And how? Through transamination into oxaloacetate, which is present in the Krebs cycle. And one intermediate which links the Krebs cycle with the urea cycle is fumarate. Understood? Now what happens is the free ammonia is highly toxic, that is produced after the metabolism of amino acids. It is highly toxic. It combines with carbon dioxide in the presence of carbamoyl phosphate synthetase 1 to produce carbamoyl phosphate. This involves the usage of ATP and magnesium ions. This carbamoyl phosphate is now going to donate its is now going to catalyze the conversion of ornithine to citrulline, and one phosphate group will be released. Now this citrulline will exit the mitochondria and it is going to produce arginosuccinate by the action of the enzyme arginosuccinate synthetase. This arginosuccinate by the action of the enzyme arginosuccinase is going to produce arginine and fumarate. Fumarate will then enter the Krebs cycle. Therefore, it links the Krebs cycle and the urea cycle directly. Now, note one thing, fumarate here is present in the cytosol, but fumarate will enter the mitochondria to enter the Krebs cycle. Do you see how compartmentalization is occurring? When fumarate is present in the cytosol, it is a part of the urea cycle. Whereas when fumarate is present in the mitochondria, it is a part of the Krebs cycle. Okay? Alright? Got it? Yeah. Understood? Now, what will happen? Now, what will happen is, this arginine is then going to convert through arginase into urea. It is going to produce urea and it is going to convert to ornithine. It is going to convert to ornithine. Okay? Understood? So, this is how the urea cycle works, and this is an excellent example of compartmentalization. Understood how? When different substrates present in different compartments enter different cycles, even in the same organ. Okay, so this is what we discussed when we started our discussion of biochemistry. The most important is compartmentalization. That is how different reactions occur inside our body. Okay, simultaneously. So this is how compartmentalization works. And this is all about the urea cycle. Also, where the conditions where urea is increased and where urea is decreased. I'll just briefly sum up. Urea is synthesized by the liver majorly, and it is excreted through the kidneys. Okay. So the conditions where urea is high, serum urea is high, and the conditions where serum urea is low. Now, one by one, we'll enumerate. Okay. Now, serum urea can be high when there is acute kidney injury. Then what will happen? And there is chronic kidney failure or disease. Then what will happen? how will you consolidate your diagnosis? The whole of the KFT will be deranged. Okay, other kidney function tests will also be deranged, like creatinine, glomerular filtration rate. All these will be deranged. Okay, uric acid. All these will be deranged. Okay, creatinine, GFR, glomerular filtrate, uric acid. All these will. Okay. Now another thing is when there is dehydration. Dehydration is also going to lead to hemoconcentration. There will be increased concentration of hemoglobin also here, and a lot of other there will be increased concentration of electrolytes also. So there will be hemoconcentration present in all the components. Okay, understood? And lastly, when the person is on a high protein diet, then also because of high protein, there will be increased secretion of ammonia, which formation of urea. Low serum urea can be in hemodilution, opposite of hemoconcentration. Okay, when the person has had a lot of water, so how will it be supported, your diagnosis? There will be dilution or there will be decrease in the concentration of most of the factors. Next is low protein diet. The person is having a very low protein diet, starvation, all these things will lead to the serum urea being low. Okay, it is going to be, how will it be supported? There will be clinical through clinical examination also you will find the person to be malnourished. There will be overall protein energy malnutrition of the person. Okay. Third is in liver disease. Liver disease, if liver is not synthesizing, is not performing its synthesizing functions properly, serum urea will be. How will you support the diagnosis? Serum albumin will also be low. Okay, and what will happen is, yeah, serum albumin can also be low here, whereas serum albumin can be low in kidney disease also, where the kidneys are not able to retain serum albumin. Now this and also clotting factors can also be affected, that are secreted by the liver, or they can be in any injury. There can be symptoms of hepatic failure as well, like ascites, like other diseases, like there can be portal hypertension, can be hepatomegaly, or all these things. Now this decreased serum albumin is going to bring me to another important topic, that is the extracellular matrix. Extracellular matrix is composed of structural proteins that are collagen, elastin, and fibrillin 1, and specialized proteins which are laminin and fibronectin. And proteoglycans. So this is the composition of extracellular matrix. Now I am going to bring your attention towards laminin. Why? Because laminin forms the basal lamina. Okay. Now since laminin forms a part of the basal lamina or the basement membrane, glomerular basement membrane. Okay. Now the glomerular basement membrane has pores. Okay, there are pores formed in the glomerular basement membrane, formed by laminin protein. This laminin protein contains heparin and heparin sulfate. Now both of these are negatively charged. What are these? What are heparin and heparin sulfate? They are, yesterday we discussed, they are glycosaminoglycans. That is GAGs. That means they are polysaccharides containing repeating units of disaccharide molecules. And these disaccharide molecules, out of these, at least one is an amino sugar. What is glycosaminoglycan? Polysaccharides containing repeating units of disaccharides, out of which at least one is an amino sugar. Understood? So there are different types of glycosaminoglycans like dermatan sulfate, keratin sulfate 1, keratin sulfate, heparin, heparin sulfate, chondroitin sulfate, hyaluronic acid, and many more. These are the most important glycosaminoglycans. Now, coming back to our discussion, the two negatively charged glycosaminoglycans, that are heparin and heparin sulfate, they are going to repel the negatively charged albumin. Okay, this albumin protein is a small protein, and it is also negatively charged, which will be repelled by the heparin and the heparin sulfate molecule present in the laminin protein, the basal lamina. Therefore, in spite of the pore of the glomerular basement membrane, larger pores of the glomerular basement membrane being larger compared to albumin, albumin is relatively prevented from being passed through the glomerular basement membrane. Why is it so? Because of repulsion of negatively charged albumin from negatively charged heparin and heparin sulfate. Understood? Hi Naman, how are you? Okay, do you have a doubt in this? We are discussing the urea cycle, to which we came, while discussing the urea cycle, we came towards the discussion on albumin, and we came back to revise our knowledge of extracellular matrix. Okay, so I think this will be it for tonight. If there's any doubt, then the session is open for doubts. The next 2 minutes you can ask your doubts, and then after that we'll conclude the session. Okay, all right guys, then please remember the intricacies of the Urea cycle, extremely important. Just look at this diagram, remember which reactions, the first two reactions are occurring inside the mitochondria, and the last three are occurring inside the cytosol. Here fumarate is present in the cytosol in the urea cycle, whereas it enters the mitochondria to enter the Krebs cycle. So the one substrate that links the urea cycle and the TCA cycle is what? Fumarate. Very, very important. Okay, understood? Alright guys, so next 2 minutes the session is open for asking questions, and then we'll conclude. One important announcement: every day I'm taking around 2 classes on YouTube and 1 class on the Unacademy app. That is the special class at night. That is regarding the MCQ sessions. Today I took 3 classes on YouTube, okay? And from tomorrow I'll be taking balanced sessions, 2 classes on YouTube and 1 class on the Unacademy app. So please, all of you, do follow. We are discussing very important topics. Tomorrow we'll be discussing about xenobiotics. Very, very important. So please pay attention and be present. Okay, not just to mark your attendance, but also to mark your attentiveness. Okay, all right. Muhammad Noor, hi. The pore size of laminin is smaller no, sorry, is larger compared to albumin. Albumin is smaller compared to the pore size. Okay, albumin is a very small molecule. It is smaller compared to the pore size, but still it cannot pass freely, because as soon as it comes near the pores, the heparin and the heparin sulfate present in the pores repel it. Okay, so it cannot be passed through the pores of the glomerular basement membrane. Understood? And that is why albumin is not present in the urine in large quantities. Okay, only a little bit of albumin can pass through. Understood? So, alright guys, then that is it for tonight. Good night. See you guys tomorrow. All the best. Revise the session. I'll see all of you tomorrow in the session on biotics and digestion, and a session on the Unacademy app on MBBS, that is MCQ bonus biochemistry sessions. That is a revision session utilizing using MCQs. Okay. Bye bye guys, then. Good night.